My name is Sverker and I represent the National Genomics infrastructure here. This is the type of gel images we get when we are about to start projects. We require, for DNA projects, we require that an agarose gel is sent in. And then what we're supposed to do when we see these is to make a call, will this work or not? It's on one hand extremely valuable information, but it's also extremely hard to make a good call about will this work for this organism, for this project, for this analysis. So typically we set the standard very high. We say that if it's at all degraded, we will fail the sample. That doesn't mean it will be a non-successful project. It just means that we can't guarantee that this will work. So we could still go on, but any reagent costs will have to be covered by the user. Some projects depend on having degraded samples. So, for instance, Pavel started talking about ancient DNA. That's one typical research question, which this is what you get. And I mean, this would be good samples for that type of question. So we do need to try and find good solutions for these types of samples. And FFPE is one such sample where we have a large benefit of being able to have good protocols to analyze these. Pavel already spoke about these points, but this is a very established clinical practice and there are loads of samples available. It's great at preserving morphology, but it's not great at preserving nucleic acids. So you see fragmentation, deamination, oxidations. You get nicks and gaps, and you have cross-linking to proteins. Also, the extractions are inefficient and sometimes biased towards where the proteins were situated. And you get very, very variable sample quality based on the age and practice of that particular sample. So this causes a bunch of different problems, all ranging from the different protocols, from fixation to extraction, library preparation, sequencing, and also analysis, since you can have anomalies in the data which you can erroneously call as variants. What we've looked at initially, and what I will be talking about today, is whole genome sequencing from these samples, so not targeted sequencing, which Pavel highlighted. And we're going to look only on library complexity as a measurement today, so nothing about analysis or other problematic areas. When we approached this, we looked at what we had, and we've already set up a low input protocol based on the Rubicon Thruplex kit. It supports input amounts from 50 picograms to 50 nanograms. We do most of our preparations automatically in order to achieve high throughput. So we can't afford very sort of complicated tinkering with single samples. We need to have stable, robust workflows where we can pipe through samples through, which is especially challenging when we're talking about these samples. But this is where we started off. And we're going to be looking at these types of curves. This is complexity curves made with the tool called PreSeq, and if you want to know more about it, you can follow the reference to the publication. But what we see in the middle here, or rather in the x-axis, we have the total number of molecules. So this would be 300 million molecules. So those are the molecules that were sequenced, and if it would be a perfect library in terms of complexity, you would also have assayed the same number of original molecules. So the unique molecules you started with are also the ones you're sequencing. Typically protocols contain PCR, so actually what you're seeing is less extent, a lower number of unique molecules. You're sequencing the same PCR molecule multiple times, or rather the same unique molecule which you've amplified multiple times. Determining what's a duplicate is in principle very simple, but in practice very hard. That's why there are multiple tools for this. We're using pre-seq. But the general principle is that when you have a high complexity library, your reads will map to the reference with a unique location like these, and when you have a low complexity, they will stack up. So here you can see that this was actually the same molecule, and then you simplify that to this was just one measurement of the original genome that corresponds to. In our test sample set, we ran an automated gel electrophoresis. So rather than looking at the agarose gels we looked at earlier, this is the same type of measurement. You have the length on the x-axis and the intensity on the y-axis. For good samples, this is a control, which is not FFPE. You see typically above, this is 40 kb. So you see that most of the genome is above 40 kb. Here we have two examples of bad FFPE samples which we processed, where you see that this is, for instance, 300 bases. So you see that most of the sample is degraded, and for the worst one we actually had a peak at 300 bases. These were the libraries that we got from our already established low input protocol. So we could see that it was very stable. We were happy to see that despite the problems associated with nicks and gaps, for instance. We saw that we got stable libraries from this. So these are duplicates. This is FFPE 5, FFPE 6, FFPE 8, and this is the second duplicate. So we see that we get good yields from the protocol. But what we're really interested in is how many sequence reads can we get out of this. So then we return to the complexity plot. Here I have, for simplicity, I've simplified it to human genome coverage, because that's what we're talking about. We want to sequence whole genomes. And so I grossly simplified the plots from 300 million reads to 30x, which is not entirely incorrect, but not entirely correct either. But it makes the plot easier to look. So I just want to bring that caveat in there. So what we see is that if we compare to the TrueSeq nano protocol for controls, we started with 50 and 100 nanogram, and the actual recommended starting amount for human genomes, if you're going to do a whole one, is 200 nanograms. So this is below the recommended amounts. We see that we would produce roughly 26x if we sequenced, from the 30x sequence. Compare that to the Thruplex protocol, where we started with 10 nanograms, we produced around 23x. So these are for good controls. This is not for FFPE. And if we then look at how it turned out for FFPE, you see the impact that these types of fixation protocols have. Sequencing more than 10x is mostly wasting reads. For the two worst ones, which we highlighted on the gel images, we see that we can produce maybe 5 to 7x from these samples. So we produce stable libraries, but we can't deliver 30x genomes. So clearly 10 nanogram is too little, and we're now working to see if 100 nanogram can at least bring it into this region in the middle, but likely it will not be up here. So that's sort of a take-home, that we can sequence these samples, but they will not perform as well as non-degraded samples. So that's what we have today. Looking a little bit forward, In the literature, a lot of protocols exist to handle ancient DNA samples, which is similar to FFPE samples in that they are deaminated, they have nicks and gaps, and they're degraded. So we're collaborating with Casper and Sten, who recently published this method in Genomics. It came out this month, actually. It's based on capturing single-stranded fragments from the library, which then can be very beneficial to preparing FFPE samples. So we did, this is preliminary data, but we did make preparations from the same complicated samples, 6 and 8. And then, so these were the old ones, and now in purple we have PCR-free library preparations of the same samples, and they turn out very, very complex, obviously because they are PCR-free. This helps. So we really hope that in the future we will be able to realize this type of setup and have that automated and be able to deliver this as a service. Right now there are a number of things to address before it's stable enough, but this looks very promising, and we also see other types of protocols coming out like this. So,
sorry, how much amount of DNA was the PCR-free input?
So I didn't put that in there because it's misleading. It's 50 nanograms that we started with here, but we didn't get enough molecules to sequence a whole genome. So currently what we're investigating is how much do we need to put in there so that we can sequence a whole genome, and we're targeting this for the HiSeq-X. So the amount we got out was maybe equivalent to half a genome from 50 nanograms, but that's roughly. And also yield is currently varying for this protocol, so we are trying to stabilize that. Another thing we're looking into on the future perspectives part, we recently had a new extraction robot installed, which targets FFPE samples. So this robot can process 12 samples in parallel in 2.5 hours. You do a centrifugation up front from your slice in a tube, and you put the tube in there, and then it does the rest. And out comes extracted DNA and RNA. Our reasoning on this is that extractions is a big part of the success rate of a project, and for these particular challenging samples, it could be we could potentially bring up the quality in the support we can give if we can also support extractions. But this is very preliminary test. We've run this once. It's data courtesy of Anders Jämt who's run it. These are RNA samples, basically just proving that it worked. But that is something we're looking into. So I'm going to end there. Just summarize that FFPE samples are particularly challenging. We see stable library preparations from our low input protocol. But they do turn out with much lower diversity than high quality samples. And for the future, we're looking into if we can bring that up with more input. And we're also running more samples to see how those turn out. We are investigating other library preps. We've also run these samples, number 6 and number 8, on the NeoPrep successfully, which we don't have data to present yet, but at least we got good yield in the libraries. We're looking into this custom protocol, and we're also trying to set up a protocol for RNA, which I have not talked about today. And the extraction robot is another thing we're looking into. So with that,